Hi guys, welcome back to Big Brain Biology. So today in this lecture video, we'll be talking about multiple allelism and Bombay phenotype. So as we all know that one gene that is going to code for a phenotype have two allelic forms located on loci of chromosomes, and that too homologous. But what if there are more than those two forms present in a population? These are termed as multiple alleles. Today, taking example of ABO blood group, we know there are some antigens that are present on RBC cell surface, and those antigens are isoagglutinogens. And these are IA, IB, and small I. But there are some antibodies also that are present and are known as isoagglutinins. Okay. So these combination of different alleles are going to give phenotype of A, B, AB and O. This concludes that IA is dominant over small I, IB is also dominant over small I, but when IA and IB are expressed, they are codominantly expressed. These multiple alleles are also known as allelic series, and the ABO blood group was first discovered by Karl and Steiner. What he did was he took blood samples and serum from his co-workers and did some reactions carrying out various mix and matches and suggesting that not just antigens but there are antibodies also present in blood groups. So if a person have a phenotype, it's going to have antigen A but not antibody against A but antibody against another antigen, that is B. Same goes for B phenotype, going to have antigen B but antibody against A. Then we have phenotype of AB, going to have antigens A and B but none of the antibodies are present. Okay. We have phenotype O, no antigens on surface but antibodies of A and B are there. So mix and matches are there, and these are important for transfusion reactions, so taking of blood from different donor, and compatibility is checked so as to take blood from different people. Suggesting that it was found that O is universal donor and AB is universal acceptor. These antigens are present on chromosome 9, and there is a precursor molecule which forms this H substance, and it has three moieties: galactose, N-acetylglucosamine and fucose. So this H substance is actually present on RBC of all individuals, irrespective of which blood group it carries. Now what happens is this H substance precursor which have galactose and N-acetylglucosamine, there is one FUT1 allele which is going to code for fucosyl transferase, and this enzyme is going to add fucose to this H substance precursor, and finally our H substance is formed carrying three moieties. Okay, if IA allele is there, it is going to add N-acetyl galactosamine to this H moiety. Okay, so this protruding sugar is there. Whereas if we have B allele, this B allele is going to add galactose to this H substance. But what if mutation occurs at the step? Which means that if FUT1 is mutated, what is it going to show? So FUT1 was going to add fucose to the H precursor, we know. So it suggests that H is not going to be made, H is not synthesized properly. If H is not synthesized properly, it is not going to protrude another antigens on its surface, and then no phenotype is being expressed. So it was seen that this woman was actually carrying phenotype of O, but genetically she was B. and it was seen through this pedigree that her outcomes was actually having B also, and she was just predicted to be O, but genetically she was not O, she was B. And this was it for this video. These are the references you can follow, and I hope you like this video. Like, share and subscribe to my channel. Thank you.